Good day, everyone. Welcome back to class. In today's class, what we want to talk about is regulation of glycolysis pathway. Regulation of glycolysis pathway. Remember, our previous class, we discussed extensively on glycolysis, its pathway, and the enzymes involved in each reaction, the enzyme that catalyzes each reaction. And a quick recap, we said we have two phases in glycolysis. We have the preparatory phase, also the energy consuming phase, and we have the payoff phase, which is the energy producing phase. And we said in total, in glycolysis, we have 2 molecules of ATP being produced, 2 molecules of pyruvate being produced, then also 2 molecules of NADH that we have in glycolysis. Now, going back to what we want to discuss today, why do we have to regulate this pathway? Remember, we discussed in our previous class that the main essence of glycolysis is to provide energy. When you consume your food and the cell breaks it down to give you the energy that you need in running or day-to-day activities of the cell. Now, why do we now have to regulate this pathway? The reason it's not far-fetched is because when we have sufficient energy, there is no point in producing more energy just to prevent wastage and to maintain homeostasis in the cell. So this particular pathway needs to be regulated. Now, where can it be regulated? And also note that when we are talking about regulation, meaning that we are telling this particular pathway at different stages of regulation that pause. Or for now, we have enough energy. I don't need you to work, so pause. So now, when the energy is depleted, we can now signal also to the pathway to resume work. So for this pathway, the glycolysis pathway, we have 3 points where this particular pathway could be regulated. Now, let's start. The first point where this pathway could be regulated is at this point, exokinase. So this is point 1. Another point where this could be regulated also is, let me use another marker for it, is at this stage, PFK1. So let's call it 2. And the last stage that this particular pathway can be regulated is here, 3. So these are the 3 stages at which this particular pathway could be regulated enzymatically. Let's take them one by one. For exokinase, exokinase is an enzyme that helps in converting glucose to glucose phosphate. Now, when we have enough energy, there is no point in producing this energy again. So we have to stop it. So and when we say we have enough energy, that means we have high concentration of ATP. So definitely when there is high concentration of ATP, there is no point for us in getting more ATP. So we have to pause the pathway. So the first thing that can inhibit this enzyme, ATP, can serve as an inhibitor. So inhibitor, ATP. Another thing that can also inhibit this enzyme is glucose 6-phosphate, which is the product of the reaction. You know, glucose is being converted to glucose 6-phosphate by exokinase. Now, when there is accumulation of these glucose 6-phosphate, it could lead to inhibition of this particular enzyme, which is exokinase. And when this enzyme is inhibited, there is no way this glucose will be converted to glucose 6-phosphate, thereby putting a pause to glycolysis pathway. So another inhibitor is glucose 6-phosphate. Glucose 6-phosphate. So these two molecules are able to inhibit this enzyme, thereby telling this enzyme to pause, inhibiting it from performing its action. So this is not, will not be able to get glucose phosphate. And if it not get glucose phosphate, there is nothing phosphoglucose isomerase will work on to give us glucose phosphate. Now let's move on. Another point where this can be regulated, we said, is at PFK1, phosphofructose 1. This is another place where this pathway can be regulated. Now, part of what can regulate this pathway, one also is ATP. Remember we said when the essence of this pathway is to give us energy, so we have enough ATP. This arrow means high. When there is high concentration of ATP, the pathway is being inhibited. Now, another thing that can inhibit this particular pathway is the concentration of acetyl-CoA and citrate. Acetyl-CoA is what we get from the breakdown of pyruvates. When we break down pyruvates, we get acetyl-CoA, and this acetyl-CoA enters into the TCA cycle to continue processes of the cell. Not only that, citrates, which is a TCA cycle, when there is accumulation of citrate also, because those are the entry point into the TCA cycle, when there is accumulation of citrate also, it could lead to inhibition of PFK1, thereby inhibiting this enzyme from further catalyzing the reaction, and invariably there will be a pause in the glycolysis pathway. So citrate could be an inhibitor, and also acetyl-CoA. So let's write. We have citrate, we have acetyl-CoA. Now let's move to the third enzyme, which is pyruvate kinase. Pyruvate kinase, when we have high concentration of ATP also, it will serve as an inhibitor of pyruvate kinase. So let's put that one first. We have ATP. Also, when we have high concentration of citrates and high concentration of acetyl-CoA, this can also serve as an inhibitor of this enzyme. So we have citrates, we also have acetyl-CoA. So now these are the three steps where it can be regulated. We've talked about the inhibitor, meaning things that can prevent them from performing their action. Now let's move on and talk about things that can activate the activators of this enzyme. For hexokinase, it can be activated by ADP. So ADP. So that means when these enzymes senses that there is ADP, meaning that there is low in concentration of ATP. So when there is low in that concentration of ATP, ATP is being used up. So when ATP is used up, it's being converted, dephosphorylation to ADP. So when there is this when the enzyme is sensing that we are having high concentration of this in the cell, so it gets activated. So it continue its reaction. Now let's move to PFK1. That's phosphofructose 1. Phosphofructose 1 can be activated by AMP. It could be activated by AMP. So also sensing, that is also telling us that there is depletion in the ATP, the phosphorus. So when there is depletion in ATP, we have, and there is abundance of AMP. This could activate phosphofructose and or instruct it to continue its work. Then also another thing that could activate this enzyme is fructose bisphosphate. Fructose 2,6 bisphosphate. That's another thing you can could activate this enzyme. Now let's move to the third enzyme, which is pyruvate kinase. Pyruvate kinase can be activated by AMP. AMP. Also, it can be activated by fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Apart from all these molecules that serve as activators and inhibitors, our hormones can also serve as activators and inhibitors. And for these enzymes, both exokinase, phosphofructose kinase, and pyruvate kinase, they can be inhibited by glucagon. They can all be inhibited. Every one of them can be inhibited by glucagon hormone. So could be inhibited by glucagon. Then all of these enzymes also can be activated by insulin. Do a quick recap of everything we've been discussing since the beginning of today's class, because you know that here we always try and make things easy so that we'll be able to understand. Well, that is what we do. Now, these are the three enzymes that we talked about earlier. The exokinase, the PFK phosphofructose 1, and also the pyruvate kinase. Now, you see the enzymes, these are the three enzymes involved. These are the inhibitors and these are the activators of these enzymes. Now, apart from this, we could also have hormonal regulation in glycolysis pathway. We could also have hormonal regulation. So for all these enzymes, they can all be inhibited by glucagon. So let's put it there. We have glucagon. Glucagon can serve as inhibitor for all these enzymes. And also on the other hand, insulin can now serve as activator of all these enzymes. Now, these are just a summary of everything that are involved in regulations of glycolysis pathway. I hope this actually be of help to you and will help you in subsequent time to be able to know your glycolysis pathway and its regulation very well. See you in the next class. Bye bye.